Greetings, students. Mr. Sanger, live from the prep room, here to talk to you about a lab that we're going to do. It involves gel electrophoresis and is called Bad Food at a Good Party. Also, this is hilarious. So here's the scenario. This lab is actually based off a real life incident that happened. You're going to investigate the source of a foodborne pathogen by looking for DNA from that pathogen, and you're going to use gel electrophoresis to compare DNA samples. You're going to actually get to do the procedure for gel electrophoresis and play with some kind of fun toys. So the scenario was in the year 2000, there were 406 cases of foodborne illness across 10 states, mostly along the West Coast. Here's some entertaining images of foodborne illness to really bring that home. This is my personal favorite. Stool sample analysis revealed that the pathogen involved was the bacterium Shigella sonnei, which is the third leading cause of foodborne outbreaks in the United States. In trying to sort of trace back the source of the contamination, it turns out that that 5 of the sick people had all attended the same party. And so from that little clue, they traced the source back to the 5-layer bean dip, delicious but deadly. And so the company that manufactured the dip made a voluntary recall, the outbreak ended, problem solved. However, researchers still needed to pinpoint the precise source of contamination, just for future knowledge to make sure that we address whatever the fundamental cause was of this. And so since this bean dip had 5 layers, really it could have been any of those 5 layers: guacamole, salsa, cheese, beans, or sour cream. And so as part of your pre-lab, you're going to read a little bit of this information to kind of get a sense of, the conditions in which each of these ingredients was prepared and stored and things like that. So they started by simply trying to isolate individual bacterial cells from the contaminated dip. And it turns out that that didn't work. Basically, you just put a little Q-tip in the dip, you swab it onto a Petri dish, and you hope something grows. They couldn't get the Shigella from any of those. And it turns out that Shigella can cause infection at extremely low doses. And there simply wasn't enough bacteria in these bean dip samples to isolate and grow. So this effort failed. They were not able to identify which layer of bean dip had the Shigella by trying to just grow the Shigella. It didn't work. So instead they said, hey, let's look for the Shigella DNA instead. Now, what technique would you use to amplify a very small of DNA into a larger quantity that could be studied? Hopefully you know the answer is polymerase chain reaction. Remember, this is this technique where you separate the 2 DNA strands, you add some primers, and you copy those strands. And you repeat the cycle so that 1 becomes 2, 2 become 4, 4 become 8. So maybe we could use this to amplify whatever DNA might be in that sample, and then we could look for it. So they looked for 2 specific genes that are found only in Shigella. The genes are called IPAH and MXIC. Those names don't matter that much. So basically, they use these DNA sequences to make the primer sequences. So the hope is that you could have the primer attach at the beginning of these sequences, and then that would amplify that sample, that particular strand, if it was present. They did that, and basically the PCR products of the Shigella DNA were 2 fragments. One fragment was 175 base pairs long. That fragment was from the IPAH gene. And then another fragment they produced with this was the 1,000 base pair sample from the MXIC gene. And these sequences were called the Shigella reference standard, meaning these are known fragments that would arise if Shigella was present. So now we have a way to get these fragments produced through PCR, but now we need to separate them and identify them. And so what technique comes in there? That's where gel electrophoresis comes in, because that could separate this 175 base pair segment from the 1,000 base pair segment. So the gist of the technique and the approach that they use: 1, they use PCR to amplify any Shigella DNA samples that may be present in each layer of the bean dip. 2, you separate the PCR products by gel electrophoresis. And then 3, we see if fragments in a particular layer of bean dip matches the fragments in that Shigella reference standard. And if the fragment sizes match, then that tells us that these fragments are in that layer of bean dip, and boom, we know Shigella is in that layer, not labor. So one final complication, control groups. We need a positive control. It turns out that some molecules in food inhibit the polymerase chain reaction, causing it to fail. So if no PCR product is found in a sample, it could mean one of two things. One, it could just mean that the Shigella is absent from that sample. Or 2, it could mean that the Shigella is present, but PCR failed. So we need a way to tell the difference. And so what happened is researchers added a lab strain of Shigella to all the samples prior to testing. And that lab control sample had this extra 1,800 base pair fragments. So they're literally adding this into each sample before testing. And so because we're adding it to there and looking for it later, we call it a positive control. A negative control is where you don't give something a particular treatment. A positive control is where you add something in and look for it later. So we're basically adding this 800 base pair fragment and looking for it later. So if that 1,800 base pair sample is present, it means that PCR was successful and we can count on our results. If that 1,800 base pair segment is not present, then we know that PCR failed and that our data is essentially meaningless. Okay. Now, yes, we have a positive control. We also should have a negative control, which is a PCR product of a reaction mixture with no DNA present. And so in that case, should we see any bands on our gel if there's no DNA? And the answer is no, we shouldn't. And if we do, then that tells us there was contamination present. So both a positive and negative control are important for this particular one. Okay, so your task for the pre-lab is you are going to choose up to 9 samples to test by gel electrophoresis. And so you get 9 wells and you get to add 1 sample into each well. So what you're going to do with your group is you're going to read through this list of possible samples and you're going to decide what should we test. We only get 9. What ones should we use? And this is, kind of realistic because budgets are limited, time is limited, and so oftentimes there's constraints. You can't just test every little thing you might want. So you're going to have to make some choices here. So let me just highlight what your choices are. The first 2 options are what are called marker standards, which are simply fragments of known length. So this first one has 5 fragments of 100, 300, 500, 1,000, 2,000 base pair sizes. And so what that tells you is, if you have, say, 1,000 base pair sequence, it will indicate how far that sample would migrate on a gel. So it kind of gives you a key, so to speak, for how long each of your little fragments are. The 1 kilobase ladder is the same thing. This contains 15 fragments ranging from 1,000 to 15,000 base pair. So you'll need to decide, do we need one of these? And if so, do we need one, both? Is one better than the other? What should we do? The next 3 are the controls. That Shigella reference standard. This is what this is like the 2 targeted base sequences that we're looking for. So then there's that positive control that we're sort of adding in, and then a negative control with no DNA at all. The last set are the bean dip samples. So 5L stands for 5 layer. That's all 5 layers together. Then you got your cheese, your sour cream, your bean, your salsa, and your guac. So again, you're going to have to choose 9 and figure that out. So the rest of this is about the actual procedure you're going to be carrying out. So on lab day, the first thing you're going to need to do is you are going to need to prepare a gel. And so to do that, you're going to need this plastic cast here. And you can see this little plastic piece comes out, and these make the wells where you're going to add your DNA. And so these slide into this little tray, and in that tray is this little glass thing where it's actually plastic, where you're going to pour the gel. So what you do is you're basically going to get, I'm pointing like that, that's not helpful. Right here, you're going to get this little, I don't know, Jello cup thing of the gel, and it's solid chunks. And so we're going to put these in the microwave. We're going to microwave them for a little bit, a few seconds, very short amount of time, and then it's going to melt that gel. And then you're simply going to pour it into one side of this tray. And that liquid jello-y stuff it's going to start liquid, but then very quickly it's going to solidify into a solid. And because this little comb is in there, it's going to make wells within that solid mass. So first thing when you get into class, we're going to get to the microwave. We're going to pour the gel so that this thing can start to solidify and harden while you're getting ready for next steps. Then you're going to transfer the gel to your gel electrophoresis chamber. So you're going to have a device that looks like this, and inside, whoops, that fell. This is where the actual tray goes, gel goes. And so there's this black piece at the bottom that falls out, and there's little lines where the wells go. And so you're simply going to take that tray. Once the gel has hardened, you're going to pull out the little gel very carefully from there, and then it's going to be this little like jello-y thing, and it's going to slide out and be slippery, so be careful. And you're just going to carefully transfer that into here, okay? Then what you're going to do is you're going to pour buffer solution into it. The buffer basically helps DNA fragments migrate through. It helps the electric current go through. So note that the sample is sitting in the middle here, and then there's a depression down here and a depression down there. And so you're going to pour buffer into both sides until they're both almost to the top of the gel. And then you're going to slowly pour a little bit more over to sort of cover it up. Okay. And then yeah, so then you're going to have to load the wells. And to load the wells, you're going to use a fancy pipette. This is called an Eppendorf Pipette, and it looks like this. And this little plastic piece is a disposable tip. And so you're going to put a tip on there. You're going to dip it into the DNA, draw some DNA up, and then add it to the wells. Now, the way this works, there's two buttons at the top of this, and they grip right in your hand, so your thumb kind of fits right over those two buttons. The first button, you're going to push it down until it stops just once. So you push it down, it stops. You put it into the liquid you need, and then you raise your thumb up, and that will draw out the precise volume of liquid that you tell it. And so these pipettes are set, I think you can see a 10 right here. I don't know, can you see that? And so I think that's 10 microliters, I think. I'm not sure the unit, but you're gonna draw out that precise amount, and then you're gonna put it into the well, which is kind of challenging. So they sort of recommend you kind of put your two elbows on the table and then kind of go like that to aim inside the well. And then when you're in the well, you push this down again till it stops once, and then continue pushing till it stops another. That's going to eject all the liquid into the well. When you're done with that, you push this smaller button here, which will then eject the tip. Check this out. This is kind of fun. Just see that? It just pops right out. And then you grab a new tip and you take your next DNA sample. Note that you can either add the buffer before you load your samples, in which case you're doing what's called the submarine technique, where you sort of put this below the buffer layer of liquid and then put it in there. Or you can add the DNA first and then very carefully, gently pour the buffer to like spread over the gel. Either way, you have to be careful that the buffer solution doesn't wash your DNA out of that. So you need to be pretty darn careful with that. Then to run the gel, all you do is you take this, you put it back into this little machine. It only fits in one direction. It kind of clicks in there. And then you plug this in, turn it on, and then it's going to run the gel. And what's cool is there's a dye in here that allows them to, it like stains the DNA and makes it glow, so that you can kind of leave this on there, and you can actually see the DNA separating as it goes. And it takes about 20 minutes to do this. And then once you're done, you're going to have these little fragments of DNA, and then your task is going to be to figure out, well, where, what's the result? So as I compare this to my controls, as I compare this to that Shigella reference standard, what have I learned? So that's it. Best of luck to all of us.